Alex, should we start? Good afternoon and good morning, ladies and gentlemen. So today is the second day of our workshop on cell culture technique, and the resource person is Dr. Alexander Strudzu. He is working in the Department of Neuroradiology, University Hospital, Tübingen University in Germany. His main research interests are design of novel imaging compounds for tissue-specific imaging and novel anti-cancer agents. So I would like now to ask Professor Alexander to start today's session.
Thank you very much. I will start by sharing my presentation. Okay, so I welcome you to this second lecture. Yesterday, my colleague, Dr. Sumla, already told you about the history of cell culture and the basics and methods and techniques and materials and machines. And today I want to go a little more in depth. So my topics today will be first a small introduction. Then I will tell you about the treatments of the cells. Then I will tell you about contamination and how to handle it. And finally, I will talk to you about troubleshooting, which problems are often found in working with cell culture and how to handle those problems. I want to say right now that at the end of each of these segments, I will leave a break for questions. So if you have any questions, you type them in the chat. And then at the end of each of the segments, we will make a break and I will address those questions. But now let's start with the first topic, the introduction. First, what is the purpose of cell culture? Why do we do it? First, the cell culture can be used as a model for the corresponding organ. Like we have cells from a specific organ, and within some limits, we can see these cells as a model. And the second thing we can do on the cells is that we can investigate cellular mechanisms, which we cannot, or much worse, do on the full organism. And we can do this on the genetic level by analyzing the DNA, the genes of these cells. We can do this on a transcriptional level, see which genes are activated, which messenger RNA is produced in which amounts. And last, on protein level, where we analyze the proteins that have been produced and that are present in the cells. The third use of cell culture is as a bioreactor to mass produce biological compounds, biological agents in a semi-artificial way by growing them in the cell culture dish. So first, the organ model. One big example using these cell culture as organ model is how a lot of the metabolic pathways that are present in the mammalian liver has been examined, where scientists isolated primary hepatocytes from rat livers and in big studies tested all kinds of foreign compounds like xenobiotics to study the pathways. They would feed these primary liver cells with all kinds of substances and then analyze the resulting products of the metabolizing of these substances. Second example of using cell culture as an organ model is like in the study of the brain. You can replicate the formation of neuronal networks by having these neuron cells in culture. And I have found this little video where I can demonstrate this. You see, in the beginning, there are a few connections and the cells are spread apart. And as the time lapse continues, you can see a real web forming of cell-cell connections forming the neural networks. And finally, the most common example of using cells as an organ model is in the cancer treatment. We have several, numerous various cancer types, depending on which organ the cancer stems from. And in the experience of the physicians, every cancer is different. So they require different treatments. And to study these differences, an approach is to excise these tumors, what is usually done in tumor surgery. And then the scientists can make cell cultures from these tumors and study the exact characteristics of the cells and also study the effects of anti-cancer compounds and anti-cancer treatment.
ये भी हो रहा है हाँ तो 1 घंटे का भी बनना खत्म होने वाला 1 घंटे का.
The next topic Okay, the next part of cell cultures and what they are used for is investigating cellular mechanisms. If we have these cells from different origins, then we can study the genetic makeup and compare the genetic makeup of different cell types and also especially different malignant cells. And with the modern developments, now what has come up is these big DNA arrays where we can take the DNA extracts from different cells and study thousands of genes in one investigation and compare the mutational status of these genes to see if there's a correlation between genetic mutation and specific diseases. The next possibility to study the cellular mechanisms of on these cells are proteome studies, where we extract the proteins from the cells and then study the protein makeups of different cells, and not only the amounts of proteins and the types of proteins that are made, but with the modern methods we can also study post-translational modification like phosphorylation or glycosylation, and in this way get some insight into which modifications are present in which cellular in which cell makeups and in which configurations. The next application is the transcriptional analysis, where we can study which genes are activated in these cells and study which proteins are expressed in this way. And this can vary very much depending on cell type, depending on which cycle stage the cells are in. And in this way, we can get an insight on the difference between the cells in their native form, in their activated form, or if there's some malignant transformation, what genes are activated in the case of cancer, for example. Finally, we can induce genetic changes using genetic engineering. If we have a candidate gene where we think and believe that it is involved in some cell cycle processes, or because I study cancer, my main approach is if we have a gene that we believe is involved in the initiation or the progression of cancer, we can study that gene specifically by using genetic engineering to knock out these genes in the cells. And then these genes will not be produced, not at all. And then once we have this knockout cell line, we can compare this cell to the native cell and also its reaction to external influences like substances or nutrient deprivation. And the other way around, again, if we have a gene that we're interested in, we can transform or transfect these cells and make them constitutively produce these genes in a big, these proteins in a big amount. And again, once we have an excess of these proteins in the cells, we can study the reaction to external influences like stress or substances and see if our theory on the involvement of these genes in certain processes pan out. The next application of cell culture is as a bioreactor. As I mentioned in the summary, to mass produce biological compounds. And this is done either by genetically modifying cells to make them produce human or mammalian proteins. Examples for this are growth factors like the hormone erythropoietin or cytokines, or we can have them produce monoclonal antibodies by making hybrid cells of B cells with tumor cells. And to be mentioned in this case, to facilitate easier mass production, the cells used in these applications are non-adherent cells. They're cells in suspension. The next point where we produce compounds using these cells is for vaccinations. A lot of the attenuated virus strains that are less infectious or not infectious, but still confer immunity to the people infected, are grown in cell culture. And with these examples that I have mentioned, these are specific viral vaccines that are used in humans. There are more that are used in veterinary medicine, but polio, hepatitis B, measles, influenza, and mumps, they are specifically grown in cells and used to vaccinate humans. Yes, one more thing with growth factors and proteins and cytokines that are grown. In some cases, like for example, insulin is also a mass-produced protein that is produced in culture, but insulin is so simple that it can be produced in bacteria, and insulin is also produced, so insulin is produced in bacteria, but for some examples, these bacteria cannot confer the post-translational modifications like phosphorylation or glycosylation that are required for the function in some more advanced hormones. This is why in the cases that I have given here in these examples, we need the mammalian cells. Okay, let's get to the next point. The next point is cell therapy. These are the cases where the cells itself are the product. Like for stem cell therapy, the embryonal stem cells are multiplied in cell culture to be then implanted into the patients to try some novel treatments to regrow nervous cells, for example, or in cancer treatment, where the bone marrow of the patients some bone marrow of the patients is removed before radiation therapy and then multiplied in culture. And then after the therapy has been performed, these bone marrow cells are reimplanted into the patients. And the final example is where whole tissues are grown in culture. These are either for partial organs like liver or kidney, where then these tissues can be used to replace damaged sections in the actual organs, or things like cartilage or skin, where in patient with joint degradation, where the joint cartilage has been used up by wear, and now there's no cartilage left, where they're just bones on bones, and of course with great pain involved. Then some cartilage from other sources will be extracted, grown in culture, and then injected into those joints. And these newly grown cartilage cells will provide some easing of this pain. And also lab-grown skin, for example, for burn victims. And this concludes the introduction part, and I want to ask if there are any questions.
No questions yet, Dr. Alexander.
Okay, thank you. Then we proceed to the treatment of cells. In the treatment, we differentiate between several different types of treatment. The first treatment is where we remove our cells either from the suspension culture or from the adherent culture to proceed to other experiment. Here we differentiate between collection, where we want to just keep the cells, maintain the cells, and prepare them for other experiments or for cell culture maintenance, and the harvesting, where we're going out from our cell culture for some analytic methods. The next is the treatment to induce a reaction, like if we expose our cells to some stimuli, which can be physical or chemical. But this is like just the first step, and the analysis will come in the second step. And third is the treatment for the analysis, which was either in one step or like directly the analysis, or in a secondary step after the reaction has been induced in the first step. And I'll take these one by one. The first part is the collection and harvesting. The collection is where the cells remain intact. This is for cell passaging or seeding or freezing, like seeding if you are preparing for an experiment and you need a specified density or need the cells in specific cell culture dishes for the subsequent analysis. In this case, for the cell collection, we have to be very gentle with the cells because they're supposed to remain vital. So we detach the cells either by trypsin or with the scraping. And for this, you should have seen the videos yesterday. And what's important at this stage is to minimize the stress on the cells by making sure to use isotonic media, do not give them any osmotic stress, and be careful and gentle with the centrifugation to not expose them to too much shearing force and too much of this centrifugal force. Then in the harvesting, as I already mentioned, this is in preparation for the further processing. The cells that remain intact are for example, for flow cytometry or FACS. And in this cell harvesting, there is fixation or lysis. In the case of flow cytometry, the cells are fixed with formaldehyde, so there's no further degradation, especially of the extracellular proteins, because we need those proteins either for staining or for receptor binding. And for either we use the flow cytometry in the processing of the cells, or we can extract the cellular compounds, either the proteins or the nucleotides, being the messenger RNA or the RNA in general, or the DNA. And in this case, we use lysis. And depending on which cellular compounds, we use different lysis methods because our cellular compounds are differently susceptible to degradation and denaturation. For example, if we're aiming to go to the proteins, we don't want to denaturate the proteins. So we have several different options to break the cell membranes and open the cell cytoplasm. We can use either purely mechanical methods like the high pressure homogenizer or bead mills, or physical methods like cavitation, ultrasonication, or we can use detergents that will disrupt the cell membranes and make them porous so we can get to the contents of the cytoplasm. Or we can do alkaline lysis, and this alkaline lysis will completely degrade the phospholipid membranes by splitting the fatty acids from the glycerol. Here I have given you example of the, of the purely physical methods. On the left, you have the bead mill. These beads of either steel, ceramic, or glass are in the container with the cells, and this container is then rolled, and the beads will cascade up and then fall down and crush the cells between them. One problem with the bead mills is that after the lysis of the cells, you have to extract your sample from the beads. Then on the right, we have the high pressure homogenizer. In this diagram, you see on the right, you have like bigger fragments. I found this picture which elucidates best the process of the homogenation. In this example, they did not use any cells, but it is the same for these droplets and as well as for the cells. You come with the high pressure from here, and through these thin slits, the cells are put under stress. And when the stress is relieved here at the edges, these four effects, turbulence, local cavitation, shear stress, and impact stress occur. And these will break up the cell membranes and make the fragments much smaller, as you can see here. Next, we're coming to the treatment. Treatment to induce a reaction that we will then in the second step analyze. The one main physical treatment that is used is radiation, either with ionizing radiation, for example, with a beta radiating cobalt-60, or what is more commonly used to induce like cell stress and cell damage is UV radiation. For example, if you have a tumor cell, or rather one type of tumor that is less susceptible to radiation treatment, like for example, it has been found that brain tumors, radiation treatment is not that successful with those. So there have been studies on brain tumor cells with this beta radiating cobalt-60, where the scientists then studied what is the cellular makeup that confers this resistance to radiation treatment in these cells. Then a standard process of inducing cell damage by physical treatment is the UV radiation. I have given you 3 examples of UV radiators here. Here a lineup where you can put several cell culture flasks and then radiate them. Here just a simple UV lamp. And here a very complex machine where you can exactly dose the wavelength of your radiation and the amount of radiation that each sample gets. And this is done to study the cellular reaction to the induced cell damage, which processes are activated, to study repair process or mutation or programmed cell death, like apoptosis. And at this point, I can mention another physical treatment that you can do with cells, which would be temperature variation, but this is a little moot because it is well known what happens with temperature treatment. If you go beyond 42 degrees centigrade, your cells will denaturate and die. And if you go lower than the optimal growth temperature of 37, the metabolism will slow down. But until you reach the freezing point, you won't have any adverse effects other than slowing down the metabolism. Okay, now we're coming to the chemical treatments with substances. The most obvious ones are, because you're keeping your cells in your cell culture medium, is to change things on this cell culture medium, these things being either the buffering solution, like which type of buffer you're using. You can do slight alterations with a pH, but be aware that most cells grow best at the neutral pH. Or you could change the nutrient makeup, change your glucose concentration, maybe see if some cells grow better with more of a certain amino acid, or add or withdraw hormones. This all can be studied and seen the effects, but what is more often done is to test foreign substances on cells and see their effects. The main thing we're looking at this is the cytotoxicity. If these substances kill our cells, and should the substances not kill the cells, we can study what other effects there are. If there are any effects on the cell proliferation, cell growth rate, or if there are other influences on the cell regulation. For example, a substance that might not be acutely cytotoxic might cause apoptosis, and in this way cause a delayed cytotoxicity. Or on the other side, if it could bind to some cytoplasmic or membrane receptors, activate some transcription factors, and increase the growth rate by 50 to 80%. All these things are not known before we test the substances. So in this way, one big field of research is testing foreign substances newly purified and found and extracted substances on cells and study the effects. Next, with these application of substances, we have the problems that we have to get these substances onto our cell culture. And obviously, because we are bound to have the cells in our growth medium, we have to also add these substances to the cell growth medium. And with some substances that are not very hydrophilic, we have the problem that we have to get them into solution. And for this, we use emulgators, like I have the examples DMSO or ethanol. And the advantages of these two is that they are auto-sterile, so no contamination, no bacteria will grow in them. And you can just add your pre-dissolved substances and then dilute them in your growth media. However, because they are auto-sterile, speaking, they will kill all the bacteria that grow in them. They will have the same toxic effects on your cells. So you have to be mindful that you need to dilute them very much so you don't have an adverse effect on your cells. And if you're running studies with compounds that you have pre-dissolved in these emulgators, be aware that you need to run a control of cells that are only treated with these emulgators at the same concentration to exclude any effect of these emulgators on your cell culture. Then for compounds where you don't have the problem with the emulgators, you still have the problem that when these compounds are prepared and purified, they are not used in a sterile environment. So you have to make them sterile. Otherwise, you are bound to bring an infection into your cell culture. And the most commonly used means for this is sterile filtration. Another possibility would be to autoclave these solutions. However, some compounds are not that stable if you're heating them up to 121 degrees centigrade. So you would use these sterile filters. And then the analytics in the cell culture. This is done either after previous treatment or the cells themselves are analyzed directly. And for these analytic methods, there are specialized dishes. I have given you a few examples here, these dishes for the ELISA readers, or here cell culture microscope slides where you can grow the cells directly on the slides. And we differentiate in here with the fixed cells or live cells. In In the live cells, we have this case that we want to either study the processes as they would occur in the actual, let's say, organisms, because we're using the cells as a model for the organs, or we need some parts of the cell process that are only present in living cells, like, for example, for the proliferation test, that we will come to later. Examples for analysis of fixed cells are the immunostaining or other staining methods. Now I'm giving you some examples here. The immunostaining is where you have your cellular target in your fixed cells. Like, for example, here you would have the alpha tubulin. And then there are two methods. Either you have direct detection, where your dye, your imaging dye, is directly on the detection antibody, or you have the secondary detection, where you have your primary antibody that is just specific for your target, and you have a secondary antibody that is specific for the type of antibody that you have used in the first step. And in this way, by using antibodies from different species, like, for example, you have rabbit, you have mouse, you have goat, you can also have rat, you can modulate your antibody makeup that you can have several different targets in one picture. Like, for example, here the Ki67 and the alpha tubulin. The DAPE is just a DNA stain, and here you have the nice overlay. And then here on the right, we have an example of the classic hematoxylin eosine staining, the HE staining, which is done in blood analysis as a standard. And have here the big granulocytes. So these are examples for fixed cells. Then we come to the live cells. And with the live cells, we have this cell proliferation test, which is done with this either MTT or XTT, where with an enzyme that is produced by living cells, we have the activation of, let's say, inactive for our detection form, which is yellow, into an active pharmazan form, which has different colors depending on the side chains. In the MTT, it is purple. In the XTT, it is red. So you can see here the amount of developing color correlates to the amount of living cells in these samples. And on the right cell, you have an example of live cell microscopy that is from my group. In this case, we have treated living cells with an imaging marked ligand. And we have found that this receptor for this ligand is present in prostate cells, prostate cancer cells. So these three examples, the two upper ones and the one on the left, are prostate cells. And if you look closely, you can see that this is like dot-like staining pattern. This is from the dye binding to the receptor and then being internalized. And this one image where you don't see anything, this is a brain tumor cell where this receptor is obviously either not present at all or just present in a very small amount, so you don't see this accumulation of our dye. So this is an example where you have analytics directly on your cell culture in a live cell environment. So this concludes the treatment part. Are there any questions on the treatment?
Dr. Alexander, so far we have only one question.
Yes.
Mr. Hafiz Muhammad Usman would like to know if centrifugation after trypsinization of cells to form pellet, does this have any characteristic change in the cells if we centrifuge after trypsinization?
In my experience, no. The trypsinization at the trypsin concentration that are usually used in the lab should not prove any danger to the cells. Like if it's an established cell line and it's not some primary cells which are more like sensitive, then you can trypsinize, then you can centrifuge trypsinized cells. The experience in my lab, not from me personally, but from a colleague, is that she happened to forget that she was working with the cells and trypsinized the cells overnight, and she could still recover some cells. This was way back when I was doing my master thesis. So yes, in a intact cell culture, then trypsinizing, and the trypsin should not be any danger for the cells. Did this answer your question?
I hope. Hello.
Okay. So then let us.
Yes,
Yeah.
Dr. Alexander, there's just one more question from Mr. Tariq Ayub, and he would like to know if you can briefly share your experience, if any, in cell treatment for biomedical applications. Like, for example, he wants to know response of biochemical sensors or any that include biomedical applications.
Mm-hmm. In cell sensors. At the moment, I'm not exactly understanding where this question is leading. Maybe we can pick this up at the end, where I can take my time to understand what the question is about.
Yes, absolutely.
Okay. So then we can proceed to the contamination.
Yes, please.
Okay, we have different types of cell culture contamination. With the first three, bacterial, yeast, and fungal contamination is easily visible. And so it can be, once it is seen, it can be handled and dealt with. Then we have the mycoplasma contamination and we have viral contamination. Let's get first to the bacterial contamination. If you have a bacterial contamination, then the first thing you see is that your medium becomes turbid and changes color. Interestingly, with bacteria, you have two different options for your contamination. You have either the aerobic bacteria, which will make the medium more acidic. So the phenol red in the medium will turn yellow. Or you will have anaerobic bacteria, which will make your medium more basic, alkaline, and your medium will turn pink. If you see this turbidity and the medium color change, then you can be sure that you can also see your bacteria in the microscope. What you should do is to quickly get rid of the contaminated cells to prevent cross-contamination. However, if you have some cells that are very precious and that you cannot easily replace, and you catch this contamination early before it has outgrown your cells, you can suppress the contamination to finish your experiment. However, you have to be mindful that you cannot be conclusive that your experiment has not been influenced by this presence of bacteria. But you can suppress the contamination with high concentrations of antibiotics. But these antibiotics, in the long run, they can only suppress the contamination and they cannot cure the contamination because they don't actually kill the bacteria, they just prevent them from multiplying. So if you really wanted to try and cure the bacterial contamination, you would have to go through many, many cycles of splitting the cells and diluting the cells in the hopes that once you keep the bacteria from multiplying and you keep diluting your cells, that at some point in the new dish that you will have just cells and no bacterial contamination. And since this is very difficult, it would be better if you have the possibility to just go back to your frozen stock and start your cell culture from a new sample. Next is the yeast and fungal contamination. Here again, the medium changes color and will become yellow. And also the medium becomes turbid. And in the fungal contamination, it should be easily visible if you have these moldy spots, or you can even see fungal fibers with your naked eye. And again, these cells you should discard quickly to prevent cross-contamination. At this point, I had a video embedded, but Dr. Acha requested that I show the video from the video file itself and not in an embedded way. Wait, where is it? I'm sorry, I will be right with you. Okay. Okay, can you see it now? So here we have the bacterial contamination, in this case with the pink medium, so you have anaerobic bacteria, here the yellow medium. And then under the microscope, you can see first the bacterial contamination. The bacteria will move a lot, and this will be quite obvious. Here in a bigger zoom factor, we can see the moving bacteria, and here is a culture that has been completely overgrown with bacteria, where you see no more cells, just the bacteria moving around. Now the yeast contamination, you will see these little segments, and yeah, they make like small islands of these segments, these single yeast cells. And with the fungal contamination, you have these long fungal fibers that are visible even with the naked eyes. Okay, so this was where we were. Next, the mycoplasma contamination. These mycoplasmas, the problem about them is that they are very small because they're just the very basic components that you need to have a self-replicating cell. And because they are so basic and so small, they can fit through these pores in these sterile filters. So even sterile filtration will not save you from mycoplasma contamination. And also because they're so small, they're not easily visible. One indicator that you have mycoplasma contaminations is that if your characteristics of your cell lines are changing, like most commonly it's the growth rate, that it will slow down to what you're used to. And there are two ways to detect this contamination. One is the polymerase chain reaction, PCR, for some mycoplasma specific fragments, or you could use the DNA stain, like I have shown you an example here, where you have on the left side the clear culture where you see just the nuclei being stained, and on the right side you see the nuclei being stained, but the surrounding area everywhere, these diffuse, because so small, diffuse DNA staining, which gives you a clear hint that you have these mycoplasma contamination. In contrast to the fully grown bacteria with a cell wall, these mycoplasma can very well be treated with antibiotics, and studies have found that this antibiotic plasmodiazine is effective in permanently removing over 80% of mycoplasma contamination in cell culture. Now, the final contamination is viral contamination. This is not playing a big role in the discussion of contamination. First, because it is very hard to detect. And second, because, as I mentioned here, often it is without any adverse effect for the cells. But studies have found that it is not very uncommon in cell culture, and many of the cell lines are already infected with viruses. However, there's very low chance of cross-contamination because these viruses are host and tissue specific. One point where viral infection is dangerous is if you're working with a primary culture of like an endogenous cells, human material, then you as the scientist can be in danger of infecting yourself if your cell material was carrying viruses, like for example, hepatitis. How we deal with contamination? Important is that we keep our workplace and our work techniques, our labor sterile. Don't be careless. Then use your own media, don't share them with your colleagues because then you are the only source of contamination, the only possible source. Incubators, I know this is not always possible if you have a lot of people working in the same facilities, but ideally you should have your own incubator. With the incubators as well as with the flow hoods, it's important to have them maintained regularly, and also your work area and your incubator and your microscope should be regularly disinfected. For Mycoplasma, you can every now and then test your cultures. And for the recovery from your potential contamination, it is important to keep a stock of frozen cells. So in summary, with the contamination, if you're working with cell culture, then the contamination will happen sporadically. And the best way to deal with this contamination is to be prepared and be quick to clean your work area and remove your contamination, and then start with a new clear batch of cells. So this concludes the contamination. Are there any questions on the contamination?
Yes, Dr. Alexander. Lalaine Hamel would like to know if there are any other ways apart from staining and PCR to detect mycoplasma contamination.
I would not know any.
We don't have any other questions.
Okay. So we're coming next to the troubleshooting segment. So which problems are commonly run into when using cell culture, and my suggestion how to approach solving these problems. And these problems are first recurring contamination. Second problem, low cell revival rate if you're thawing your cells. Then if you are working with adherent cells, that the cells are not attaching. And the final 2 points are slow cell growth and morphology changes, like if the cells don't behave the way you were expecting them to. The first problem, the recurring contamination, it is important to identify your source of contamination. These can be either your cell culture media or your workplace, your work area. So for your media, you can do test cultures with just your media in your incubators. And then you can test your growth media, your bovine serum, your PBS, your trypsin. Then next you can see if you have an contamination in your incubator or in your flow hood. For example, if your filters are defective, you can use these microbiological plates and place them openly in the hood and the incubator. for 20 minutes, and then you seal them, and then you keep them in the incubator. And if after 2-3 days you have culture on those plates, then you know what the source of your contamination is. Another source of contamination is that your cells have already been contaminated when you thaw them from the frozen stock. So what you can do with if you run into contamination over and over again while starting from frozen cells, you can just try a small bit of your frozen cell sample, plate them on these microbiological plates and see if something grows, and then you would know. Once you know where your contamination comes from, it's important to then remedy the problem, either discard all the media, or if your incubator was the source, then properly clean it with bleach and disinfectant. And then, for example, if the hood was just the source, you can see if the high exclusion particle filter is still intact, or if that was the source. And once you have found the problem, you remedy the problem, you fix what was causing the contamination. And then before you start your cell culture again, you should test first if you really remove the contamination. Otherwise, you would start the work, and instead of wasting 3-4 days just testing if your setup is clean again, you would also waste your valuable cells and research materials if you get the contamination again because you didn't clear it properly in the first try. The second troubleshooting target is if you have problems with the revival of your cells, like if you thaw your cells and you have no or just very few vital cells in your dish afterwards. The first thing is optimize your revival process, because these freezing media are supposed to stop the formation of ice crystals, which will penetrate the cell membranes. We are limited in the choice of these freezing media. All the available freezing media either have DMSO, bovine serum, or glycerol. And some of these, except for the bovine serum, they are harmful for the cells. So you should minimize your exposure of the cells to the freezing medium while they are still metabolically active. So once you thaw the cells, you should quickly come out from the freezing medium, and which the point we will get to later, when you are freezing the cells, you should also, once you're in the freezing medium, quickly lower the temperature. Another thing, when the cells are freshly thawed, they are very unstable. The membranes are a little shaky still. So you should avoid additional stress. Many revival protocols say that you should dilute the freezing medium in fresh warm medium and then centrifuge to remove the freezing medium. But what I have found in my experience, that it is better to just dilute the freezing medium and quickly get the cells on your dish or in your flask. And then after 2 to 4 hours, the first cells should start attaching. And at that point, you can just discard the supernatant, and this will make it much more efficient, much less stress for the cells. And personally, I have found that extended exposure to the medium containing the debris from the cells that did not survive the freezing, and the freezing medium, if we leave this overnight, the cells don't like it this much, but if you change the medium as soon as they have started attaching, this is the best. Now, the next point, if you still have problems, then maybe your cells did not take offense on the thawing, but your cells got damaged while you were freezing them. I have already mentioned that you should minimize the exposure to the freezing medium while the cells are still metabolically active. So as soon as you transfer the cells in the freezing medium, you should start your cooling and freezing rate You should quickly go, as soon as you add the freezing medium, go on ice, and then quickly go in the freezer for a half hour, 1 hour, and then quickly go into the -80 freezer from there. Be mindful not to have your samples like naked, have them on ice in a styrofoam box, because this will prevent the shock freezing that is quicker than about 1 degree centigrade per minute, because this will produce additional stress for the cells. If you still have problem, then you can try alternative freezing media. I have mentioned the 3 most commonly used ones: glycerol, DMSO, and bovine serum. And one advice that we found in our lab, some cells, after they are thawed, they like to have some cell-cell contact. So if you use too big a dish or flask for reviving, then they will be lacking this contact, and then you will have single attached cells. But because there are no cells, no other cells close, they will grow very slowly or even start dying off. We have found that some cells really like to have other cells close with which they can do cell-cell interaction. Now the next problem, if your cells are supposed to be adherent cells, but they don't attach properly. The first thing you can do, you can play around with your growth medium, use different basal medium, add growth factors, modify your glucose percentages and your FBS percentages. But the second thing you can do is that you can optimize your cell culture dishes. The usual cell culture dishes are just simple plastic, and some of these plastic dishes are not optimal, or some cells do not optimally attach to simple plastic dishes. So you can have the option of using pre-treated dishes in which these plastics are treated with some chemicals to make them more porous and make the cells easier to attach. Or if this is not enough, then you can also get coated dishes where the surface is coated with some protein, which then is easier for the cells to form protein-protein interactions with their extracellular proteins. This coating you can also do yourself. You can, for example, have some collagen and cook a collagen soup. And while this is still hot, just add a few milliliters of your collagen soup to your dish, and then leave it in the dish for 2 hours. Then discard the supernatant, and then sterilize your now coated dishes with the UV lamp, and then you have self-made coated dishes. The next problems are like 2 problems with the same characteristics and the same solution. Either your cells don't grow as quickly as you were used to or as you are expecting from the documentation, or your cells change morphology either to the way they were when you used them previously or the way they are documented in your material. Again, this can be remedied possibly if you find optimized conditions for the cell growth, either with trying different media, modifying the media growth factors, glucose, bovine serum. But a sudden change in cell characteristics are often an indicator of possible contamination. So if you have this and you have no obvious bacteria, then you should test for mycoplasma. In general, troubleshooting advice is first, nothing runs smoothly, so you will run into smaller or bigger problems. And be aware that you're not the only person working on this problem and with those cells and in this environment. So it's always worth finding somebody who is working in the same field with the same cells and asking for advice. If it's not in your own group, you can you can screen some publications and find groups that are working in the same cells, the same methods, and ask if they have any suggestions, because it is likely that if you are having problems, then somebody else has already run in those problems, and maybe they have found a solution that will fit your situation as well. And while you're talking to these other groups that are working on the same cells, you can compare what you're doing and how you're doing, and maybe get some ideas how to improve your work. Another option where you can go to for advice is companies, because these biotech companies, they also sell and work with these cells and materials, media and solutions. So they also have some experience. But if you're approaching these companies, be aware that they will try to sell you their solution, their product, and don't just blindly do what they're saying, but you rather use their feedback and their information to get new ideas to solve your problems. So this concludes the troubleshooting part. If there are any questions here, then please go ahead.
There is only one question from Professor Reza, and he would like to know how can we clean our cells from mycoplasma contamination?
This is not that difficult. The same way that you clean yourself from bacterial contamination, these mycoplasma are even more susceptible to being killed with this disinfection because they don't have the bacterial cell wall. So this is also the reason why they are really well treatable with these antibiotics, because they're not as durable as bacteria, but in return they're everywhere and it's very small. So yes, just do the disinfection of your hands and the regular disinfection of your work area and the sterile working. Don't breathe on your samples, all the things that you are usually told, and that my colleague, Dr. Sumla, should have told you yesterday, and I also believe she has instructed you well on this.
Dr. Alexander, there is one more question. I would like to know if for the slower growth rate, can we increase the concentration of FPS in the media from 10 to some like 15 or 20? And how much increase is actually recommended?
Yes, I have mentioned this in the list, that one solution for the cell growth problems is increase the FPS percentage. It is suggested not to use more than 20%. You can try more. I mean, it's no hard and fast rule, but you should be aware that at some point you are moving away from your defined cell culture medium, because the FPS, you don't know exactly what's in there. All the batches are tested for some harmful contents and possible infection, and they're also all immobilized with heat to kill contaminants. But the content of the bovine serum is not exactly classified. So with this you're moving away from your defined media and defined growth conditions. And even if you might manage to get your cells to grow better by using 25% FPS, it could well be that with your next batch of FPS, this won't work as well.
Another question?
But yes. Okay, yes, the modulation of the FPS is one of the big things that you can try. For example, in our group, we have this example of one cell line that with a lower FPS percentage, just 5% FPS, it makes really nice monolayer. And this is optimal if you want to take nice images for our publications. But in this configuration, the cell growth rate is reduced by around 20–30%. So if you just want to maintain the cells or grow the cells in preparation for the experiment, we grow them in 10% FPS. And then once we need to take images for publications, we seed them in 5% FPS. And in this way, they form better monolayers.
Before I proceed with the next question, I would like to know, is this the end of your presentation, Dr. Alex?
Yes, this was the end. I was on the thank you page.
Okay. I just have a few more questions.
Yes, sure.
Abdul Mateen would like to know if you can please let us know some of the characteristic changes that occur when Mycoplasma contaminate the cell line.
What contamination? I didn't get that.
He just wants to know if the characteristic changes in the cell line infected with Mycoplasma.
Characteristic changes. The main thing is that the cell growth rate goes down by a lot. This is what we had in our one instance where we had mycoplasma contamination. Cells that would usually take 3 to 4 days to grow from, like, freshly after splitting 10% constituency to full would now take 1 week or 1 whole week to become full. This was the main thing that I saw with the mycoplasma.
Would also like to know how to differentiate one organism contamination from the other.
Beg pardon, what contamination?
One organism contamination from the other, like how to differentiate between a bacterial contamination and the yeast contamination.
Yes, for this we can go to the video. You see here in the bacterial contamination, there's a lot of movement. These bacteria are jumping around everywhere. And they're also smaller. You can see in comparison to the yeast, which will come now. In the yeast, there's also slight movement, some vibration, but you can see the yeast, first they're bigger, and they make these specificals like these, like trains where you have the single train compartments. This is when they divide the yeast cells, but then they stay attached. And if we go back to the beginning of the yeast video, you will see some slight movement, some slight vibration here in these, but it is by far less than you have in the bacterial contamination, where they're jumping like crazy. And they're also much smaller. You can see clearly that they're much smaller. And with the fungal contamination, it's not even an issue. These long fibers, these are very obviously fungi. Yes, any other questions?
Yes, would also like to know how, is it wise to save a cell line, contaminated cell line with antibiotics? And what are the success rates in such cases?
It's not really worth it, so I can't give you success rates. I know that it can be tried, but usually the only cells worth saving are primary cells, and these primary cells, they usually denaturate after some passages, so you cannot run enough passages to get rid of your contamination. One thing you can do in primary cell lines, one thing you would do in primary cell line is that you would suppress the contamination for the duration of experiment, and then you would publish your results with the reminder that at some point you got the bacterial contamination which you suppressed, and then your results are still valid, but with a little question mark. So yes, it is usually in the cells that would be able to be saved. It's not worth saving them because you are better off if you just use a frozen vial. Go ahead.
There is another question that if a cell line is sensitive, is it wise to use trypsinization frequently?
As I already said to a question earlier, trypsinization is not a real cell stress, because all this trypsinization does is it cleaves the extracellular proteins. Trypsinization does nothing to the cellular membrane. It just detaches the extracellular proteins. It just destroys the extracellular proteins. The one thing it might do is that the cell would have to work more to rebuild these extracellular proteins once you have it reattached. But this will maybe delay your cell growth, the initiation of cell growth by a few hours if you have to have it attached first.
How important is it to have a different set of lab coat when working in cell culture facility?
This is quite important because your options for contamination are then very limited. The only contaminants that get onto the cell culture lab coat are the ones that are already present or the ones that are new. But if with your lab coat that you're moving around the premises, if you going outside, you're going in all different kinds of rooms, you just exposed to much more variable things. And really with the cell culture lab coat, they should only be the bacterial and yeast and fungal contaminants that are already in your cell culture facility present or on your person. So you can reduce your possible contaminations by a lot. Any other questions?
Professor Raza would like to know if a change in morphology of cells, what change in morphology would be associated with mycoplasma contamination? How drastic would be the change in morphology?
From my one experience with a mycoplasma infection, this was not that drastic. However, what was that the cells, they usually stretch out, become form like, or rather this is cell specific. We had at that time prostate cells that formed like little bricks or little hexagons. And after the contamination, those were more and more, like there were still some of these bricks or hexagons, but more and more cells became just very simple spindly. Like they have one, two or three extensions of the cytoplasm, but they did no longer form these nice stretched out forms.
How often should one change the media in the flask?
Well, this is why we have the indicator. You can change the medium once the nutrients run out. But yes, as a rule of thumb, every second day. If you have freshly seeded cells with a very low density, you can go 3 days, maybe even 4 days. But in our lab, we do every second day, do the medium change. And then we usually time the splitting for the weekend. So then that over the weekend, we can have 3 days.
Dr. Alexander, there is one more question. If one does not have a specified unit for cell culturing as a separate facility, what measures can one take to optimize one lab for cell culture?
One lab for cell culture. First of all, keep your flow hood as close away from the door as possible, and ideally the incubator right next to it and the microscope across from it. So there's very little movement between the hood and the incubator and the microscope, and as far away from the traffic as possible. because a lot of sources of contamination is just the air movement when along with the airstream there's some bacteria get blown into your incubator most of the times. Because the hood, when it's running, it's blowing against the airstream. Yes.
Dr. Alex, there is in African countries, because we have lots of participants from African region too, and in several countries, most of the countries, the cell culture facility doesn't existing. Like that's why we asked this question. And they always, because to develop a facility, they are always asking which type of safety cabinet they need to get.
You mean the flow hood?
Yeah, exactly.
They don't differ much. The important part is that they have this high exclusion particle filter, and the rest is like, yeah, they should have this good filter and the good laminar airflow. The rest can be manufactured. For example, a lot of these cabinets come with an integrated UV lamp, but this is not necessary. We have, for example, in our building, we have one older cabinet where that did not have an integrated UV lamp. So we just found our own solution and had our technician mount a UV lamp on a stand. And then we use this like externally. We just move this UV lamp inside the incubator for the sterilization. So it doesn't have to be all integrated. The important part is to have this good filter and the good airflow.
So what is the difference between A2 and B2 safety cabinets?
Oh, this is a technical question. I'm afraid that I'm not versed on this technical part.
Okay, so you would recommend, when someone is acquiring safety cabinets, so not only the company, but the other, specific parameters, if you can explain a little bit about it.
Again, I'm not that well versed with the technical aspect of the materials and the machines, so I really can't help you there. As I said, what's important is that the air filtration is a given. I'm really sorry, I can't help you more with this.
Dr. Alexander, I would like to know that what is, how many, there is a chance that a contamination would spread by the incubator. Like from the incubator to all of the flasks that are kept in it.
Well, it's really hard to give odds, but I would say that the chances are quite likely because you have the fan in the incubator, and if you have a contamination in the incubator, and because of the air exchange, you're usually you have either filter caps or you're not closing the bottles fully, so you have this. Or rather, with filter caps, you should be better off. But if you're doing like we do in our lab and not closing the bottles 100%, so you have the air exchange to maintain the buffering with the carbon dioxide buffer, then because the fan in the incubator moves around the air, then it is very likely that once you have a contamination in the incubator, you will get it in all the flasks.
Dr. Alexander, can I ask one question?
Yes, of course.
Okay, thank you. First of all, thank you so much for informative talk. I just want to know that which one is more efficient in both assays, MTT and XTT, with respect to cytotoxicity?
They are, from the efficiency point of view, they are equally efficient because they both uh, are targeted by exactly the same enzyme. The thing is, MTT is not that easy to handle because with the formation, it will precipitate the cleaved off pharmazan. It will precipitate and form crystals. And at a certain point, if these crystals grow, they will penetrate the cell membrane and destroy the cells. And also because they are precipitated and not in solution, if you want to quantify your results, if you want to measure in your ELISA reader, then you first have to get the pharmazan in solution again. So you have to necessarily do, let's say, medium change. You have to add solvent.
Okay.
So it's just additional work steps. This is why the XTT is much more user-friendly. It saves the scientists work because they can just add the XTT to the existing sample and leave it there for 2-3 hours and then immediately go measure the results. Other than that, they're exactly the same experiment.
Okay, thank you so much.
If I'm not mistaken, then MTT is by quite a big margin cheaper. So if you have the cost factor, then it is much more beneficial to keep using the XTT and just do this additional step in the detection.
Right. Thank you so much. Thank you so much, Doctor.
Is there any other question?
If there are no more questions right now, then I would like to go back to the questions from the treatment part. And I would like to try and understand what exactly the question was.
Thank you, Dr. Alexander, first of all, for your vast experience, sharing with us your knowledge and all. Actually, biomedical application, let me tell you a scenario a bit so that you may understand. We are trying to collaborate with the biomedical engineering department with our cell culture facility so that we could carry out some experimentation. Now, these guys are trying to develop like the biochemical sensors. We could see the cell response to specific design sensors. Maybe they want to see how the concentration of the various substances changes, and after that they are going to introduce a probe which could read the concentration directly onto the cell culture and then amplify the signal and things like that. From that point of view, the question was there that if you have any such an experience where you had like collaboration with them, and so it may be helpful for me as well.
I'm sorry, I have no experience in this case because we're pretty much, I don't know how much in the introduction you got to know about my work. I am designing these novel agents. Like I showed you one picture in the analysis part where I designed a specific ligand for a receptor that we found to be overexpressed in prostate cells. Like in this way, we are designing these ligands and label them for imaging, and then we evaluate them on the cell culture. So in this way, we're always doing direct. We're not measuring any changes in the cell culture. We're measuring the direct effects.
Yeah, it is understandable. I have went through, not through in detail, a few of your articles, and this work of like designing the probes, the fluorescent probes. It's, of course, it's very helpful in the biomedical industry. Just curious if you collaborated with them to go a next step where they could develop, where they are developing the sensor, which could amplify this signal into like more electronically expressible thing. Just curious about that.
No, I'm sorry, no, we did not get to that stage.
No, but thank you very much for that great presentation and sharing, your great work. Thank you so much.
Thank you so much, everyone. And now we come to the end of our 2 days online certificate course on cell culture technique. CG of Comstech is here with us. So I would request Professor Dr. Iqbal Chaudhry for the concluding remarks.
First of all, Alex, hello to you. It is a pity that we are not meeting physically here. We waited for you this year, but could not happen. Hopefully, soon we'll be meeting you here or in Germany. I'm also grateful to Sumla. Both of them are dear friend, Professor Dr. Alexander Stozo and Dr. Sumla Sheikh Farouk. Both of them are expert in their field. They have lovely personalities, very helping, extremely motivated. And Alex, as you can see, the house is full. There's over 125 participants, which is something really rare. Actually, what happened is that people start with large number, and as you move forward towards the end of the workshop, you lose participant. Actually, reverse happened in your workshop. There were fewer people to begin with, and as we move forward, we had more people. So that actually reflects not only the content of the workshop, which is certainly extremely important, but also the way it was delivered and your own popularity as a good teacher. On behalf of Comstech, which you know is an institution called Ministerial Standing Committee for Science and Technology in OIC member states, 57 member countries. I'd like to thank you and Dr. Sumla and my team in International Center for Chemical and Biological Sciences, Dr. Atiyatul Wahab, Ms. Kiran, and other outstanding colleagues, as well as my colleagues in Comstech for putting together a great show, something which I'm sure will benefit everyone. This is beginning of an extremely exciting era. I'm sure that this ability to connect ourselves with each other interactively would further bring knowledge together. Though we will meet in workshops and conferences, but this journey of knowledge would continue to flourish. Once again, I'm extremely grateful to all of you, and I wish you all the best. Stay safe and take care of yourself. Thank you. Dr. Sumla is online also. She must be around somewhere. I don't want to invite Sumla for comments, because then this workshop will end tomorrow. But I'm sure whatever she would say would be equally interesting. So thank you very much. Bye-bye.